Well, good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is entitled Liquid Handling Performance and Quality Assurance, Gain Confidence with Game-Changing Strategies. My name is Ryan Muse and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive and webinars work best when you're involved. So please feel free to submit your questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using the same chat panel. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Advanced Instruments who developed the content for this presentation. Advanced Instruments is a global company offering a novel portfolio of analytical tools, including Osmotech, a robust line of microosmometers to support bioprocessing and quality control, and Solentum, a portfolio of best-in-class imaging and single-cell deposition technologies for cell line development workflows and assurance of clonality for regulatory bodies. Now I'd like to introduce our speakers for today's event. Dr. Melinda Gold is a product manager at Advanced Instruments. Her educational background is in computer science, public health, and molecular biology. She has over a decade in product management experience supporting liquid handling instruments and consumables. Her education, research, and work experience have given her the skills to develop and manage products affecting life science and public health. And with over 25 years of experience in technical product training and documentation, Candy Gilman is an education and training manager at Advanced Instruments. She leads the training team's efforts for Artel's liquid handling certificate programs, which include delivery of training classes, training content, and media management. She has certified hundreds of laboratorians on pipette technique and worked with many companies to implement a robust in-house pipette quality management program. Now, before we move on, we have a poll question for audience members. This should be appearing on everyone's screen right now, and you can participate and interact with this in real time by selecting on any of the answers that you see in front of you and then clicking submit. The question that we're asking is, what is the largest source of pipette failure? Your answer options are environmental conditions, inherent variability, operator error, predictable failure, or accidental failure. We'll give everyone some time to consider your answer and participate, and then in a moment, we'll hear from our wonderful speakers about today's topic. The question again is wondering, what is the largest source of pipette failure? It looks like most of you have submitted an answer, so thank you very much for participating. Let's take a look at your results. We have overwhelmingly 100% of you selecting operator error. Thank you very much. I think we've got a hint of some information we're about to hear from our presentation. So without further ado, I'd like to go ahead and hand the mic over to our first speaker, Candy Gilman. You may begin when you're ready.
Thank you so much for the introduction, Ryan. I want to take a moment and discuss the details of our agenda today. We'll start by having a brief discussion about liquid handling quality assurance with a focus on the importance of pipettes and their operators. Next, we'll review information about automated liquid handlers and the impact of various liquid class properties. We'll tie together those worlds of manual pipetting and automation with an overview of several standards and what they have to say about calibration, verification, and operator trainings. We'll review several case studies that integrate our systems and services into their liquid handling quality assurance programs. And finally, we'll close with a live question and answer session. Let's go ahead and get started. We all know and understand the importance of correct volumes in a laboratory process. Now, whether you're testing a single channel pipette in Boston, a multi-channel pipette in Brussels, or an automated liquid handler in Beijing, having a robust standardized process to evaluate those volume transfers is critical. Now, verifying volume delivery has multiple facets. It's not just about checking your pipettes, but assessing your pipette operators. It's not just about improving your automated liquid handler performance, but also about optimizing liquid classes on your ALH, all while balancing various requirements and standards that may be related to those very same topics. So let's go back to the beginning of every assay, the manual pipette. Now, we performed a study on a Boston-based pharmaceutical company who over the course of 5 years experienced a 24% as found failure rate. Now imagine 1 out of every 4 pipettes was simply not functioning. For this company, their 24% failure rate was caused by 2 key items: variability in pipetting technique and the need to increase their pipette calibration and verification cycles. In our experience, we know that operators cause the largest source of variability when it comes to pipette performance. After all, we are non-standardized beings attempting to perform a standardized process, and that can sometimes be difficult. Pipette operator variability is often the elephant in the room. If you walk through your laboratory, are you able to see visible differences in how operators are handling and using their pipettes? Now imagine if you are able to see those differences, consider how they may be impacting data or product. Now, this data illustrates a compelling story of just how much variability pipette operators contribute. Now, through the years, I've had the pleasure of certifying hundreds of analysts and scientists, some that have even been on the bench for decades, and many who have never received formal pipetting technique training. I will tell you, even the most seasoned technician gleans a nugget or two of critical information that helps significantly improve their data. As you review this slide, think about where your pipette operators might fall. We provide many resources on this topic of manual pipetting and pipette calibration, including posters and lab reports to help guide you along your liquid handling quality assurance journey. If you are interested to know more, reach out to us. We're happy to help. So now that we've talked briefly about pipettes and their operators, let's swap to the automated side of the workflow. Many labs are implementing automated liquid handlers, or ALHs, to help provide higher throughput and greater reproducibility in their intricate procedures that might involve complex liquids and multi-step assays. Now, we wanted to take a look at what's really the impact of some of these small changes. What's really the impact of something like 2 microliters? We took a model high throughput screening assay with key components, reagent 1 and reagent 2 compounds, that were supposed to be delivered at 25 microliters. We intentionally varied those volumes by plus or minus 2 microliters to measure the impact of the potency, or the IC50, at the conclusion of the test. And on the bottom right-hand side of the screen, the measured IC50 had a very widespread min to max just by adjusting those reagent volumes by 2 microliters. Now, this slide provides a bit more detail on how adjustments on an ALH can change your results dramatically. On the left-hand side, you'll see various robot settings that were adjusted, with iterative testing performed throughout every change. And in this particular case, the liquid class for the serum sub-solution was finally dialed in after making 8 different sets of changes. We began with a relative inaccuracy of minus 6.9% and a CV of 2.7, and ended with a relative inaccuracy of 0.8 and a CV of 0.3. So some of those same settings we adjusted on the automated liquid handler in that previous slide are also mentioned in our ALH troubleshooting poster. This easy-to-use quick reference provides insight to some of the basic items to consider when automated results are not ideal. So understanding how pipettes and ALHs function is only part of the equation. Ensuring device and operator performance while satisfying standards or various regulatory requirements can at times feel like a daunting task. There are many recommended practices contained in ISO standards, clinical guidelines, and various federal requirements. Now in the next slide, we'll summarize their information into one space. Now, a robust calibration verification program often contains details about frequency, volumes, replicates, and tolerances. In summary, we recommend calibrating quarterly or at least every 12 months and performing routine tests every 3 months. Make sure that all multi-channel devices have each channel tested and that adjustable volume devices are tested at low, mid, and high volumes with 10 replicates per volume. Now, with regards to tolerances, they can take shape in many forms. Consider your process tolerances or establishing a systematic error of plus or minus 2% with a random error of less than or equal to 1%. Now I'm going to go into a little bit of detail about what is included in ISO 8655, as it provides quite a bit of guidance for pipettes. Now, not all of the sections are relevant to our discussion today, but let's talk about the ones that are. In Part 1, key terminology is defined, as well as user requirements and recommendations for calibrations, testing, and routine tests. Parts 2 through 5 and Part 9, which is brand new, addresses manually operated POVA and their maximum permissible errors and allowable measurement uncertainty for pipettes, burettes, diluters, and dispensers. And in Part 9 again, which is new, laboratory syringes are also now included. Next, we'll review the new ISO 23783 standard for automated liquid handling systems. Now, in Part 1, it sets vocabulary and general requirements, specifically requirements for calibrations and routine testing, as well as calibrations and testing with liquids and liquid classes that are representative of real samples. Part 2 describes test methods and includes the dual dye ratiometric method. Part 3 describes statistics and reporting, specifically descriptive statistics for calculating volumetric errors and reporting requirements for test results. Okay, so now I'll turn it over to my colleague, Dr. Melinda Gold, who will discuss how our products and systems can help support your liquid handling efforts.
Thanks, Candy. Now that you've gotten us through the groundwork, I can provide a little more insight into how advanced instruments can help facilitate conformity to the standards and recommended practices with the Artel technology and services. Artel has been around since the 1980s and has been widely recognized as the global leader in liquid handling quality assurance. Our products and services provide standardized quality to many laboratories all over the world. Artel's technology is based upon dual dye ratiometric photometry, which simply means that the test liquid contains two distinct dyes, one blue and one red, with absorbances measured at 520 and 730 nanometers. The concentration and ratio of the two absorbances are used to calculate the dispensed volume. Inherently, the method does not require stringent environmental controls and therefore is very well suited for low volume verification. I'll show you a quick animation to accompany this explanation on the next slide. The technology and solutions are part of an unbroken chain of traceability from 5 mils all the way down to 10 nanoliters. In this example, you can see that we begin with a vial filled with a blue dye. Absorbance is measured at 730 nanometers. Then the sample is added, which is a red dye. This is the volume of interest. The two solutions are mixed together. And finally, the absorbance is measured at 520 nanometers. The software then calculates the volume of interest. The MVS, or multi-channel verification system, is used for verification of virtually all automated liquid handlers and multi-channel pipettes, with a testing range that can extend all the way down to 0.001 microliters, 100 picoliters, with results achievable in less than 10 minutes. The system can be used with aqueous and non-aqueous solutions to mimic your specific volume transfers that match your actual assays. Because the system uses the dual dye ratiometric photometry I mentioned earlier, the need for environmental controls that are standard with gravimetric measurements is eliminated. Now, I'm going to dive into the first of 4 case studies that I'll discuss. The first one is specific to the MVS. This case study exemplifies how the MVS ensured accurate results for high-stake patient samples, specifically with regards to the first genome-based test for breast cancer. Today and every day, samples from thousands of patients, women, from around the world are sent to laboratories, sometimes in ultra-low volumes, for testing for breast cancer. Samples are virtually irreplaceable, and results need to be timely, accurate, and precise. The stakes are high. If a sample is used and the incorrect results are generated, there may not be another chance, and most certainly the clock is ticking since treatment can only commence once a diagnosis is made. Delaying a diagnosis can delay treatment and potentially lead to preventable fatality in the most extreme cases. In this case, the company doing the testing employed the use of the MVS to ensure accurate results for the patients are guaranteed. The next solution for liquid handling quality assurance is the flagship product in the Artel portfolio, the PCS, or the Pipette Calibration System. The system enables calibrations and verifications from handheld pipettes from 100 nanoliters up to 5 milliliters in less than 3 minutes, and for pipettes larger than 5 milliliters, a balance can be used and integrated with the Artelware software so all data and records for liquid handling devices can be kept together. The system supports operator competency assessments and is ideal for operator training. Now, for the second case study we'll review, one specific to the PCS. In this case, the PCS helped facilitate compliance to regulatory standards in a large reference laboratory. Reference labs offer a wide range of diagnostic tests that require manual pipetting steps. Many of these labs work under strict cost constraints and have very high standards for quality, productivity, and regulatory compliance, and this lab was no different. The critical nature of diagnostic testing demands reliable results to ensure positive patient outcomes while following clear regulatory guidelines. This lab uses the PCS to ensure all their pipettes are functioning all the time by implementing routine calibrations as well as ad hoc verifications. The software provides a database of calibration records, which facilitates their regulatory requirements. The PCS is a must-have for this reference lab and many others like it. In addition to those systems, the MVS and the PCS, there are services that employ the same technology. In the case of these services, laboratories that have strict regulatory requirements and/or minimal staffing to allocate to quality control methods will choose these services. The specialists that perform these services will use the MVS to perform volume measurements on all your critical liquid handlers to avoid downtime and help you stay on schedule. They can keep your workflows moving forward by ensuring that your automated liquid handlers dispense the correct volume. The ALHS service is fully customizable. The specialist that comes on site will work on any or all of your automated liquid handlers. They will discuss your specific target volumes of interest, perform volume measurements on all your liquid handlers, teach tip configuration best practices, help you rationalize the number of replicates needed, troubleshoot and optimize your entire process from beginning to end. The accredited service, the ACS, is the first on-site ISO 17025 accredited calibration service for automated liquid handlers. After that service is delivered, the specialist will produce test reports for each test, which include well-by-well and tip-by-tip measured volumes, summary tables that can be opened in Excel, and of course, calibration certificates from our ISO 17025 accredited team. Now, for the third case study, I'll set the stage with this. The genomics field is growing and enabling personalized medicine. Personalized diagnoses involve a myriad of automated and advanced techniques on a variety of automated liquid handling platforms, and again, the stakes are high. The repeatability of results are essential, and it's important for labs like this one to validate their assays. The ALHS Artel Liquid Handling Service did just that and validated the personalized medicine diagnoses by ensuring repeatability and accuracy on a variety of ALH platforms, which is just what this lab needed. But it's not only automated liquid handlers that need to be assessed and verified. In the case of manual handheld pipettes, operators should be assessed and also trained, and their techniques should be standardized. Candy talked about the importance of this earlier and pointed out some data that showed the efficacy of training in a before and after diagram. That diagram was actually generated with data from trainings that she conducted. And even though pipettes are at the heart of probably every life science lab, many laboratory analysts have not been formally trained on proper pipetting technique. Data quality starts the minute you pick up a pipette, but ensuring accurate liquid transfers requires proper pipetting technique and liquid handling quality management. Therefore, we use the same technology to provide that feedback and training in two specific courses. The first, the Pipetting Proficiency Certification, is a one-day, hands-on pipetting technique seminar that will teach you pipette mechanics, proper technique, how to identify causes of pipette failure, how to recognize ergonomic risk factors and sources of stress, and, depending on the needs of the lab, additional modules such as multi-channel techniques can be included. The second course, Pipette Quality Management Certification, is a two-day hands-on training that will train you to establish and maintain a robust QA/QC program for your lab's pipettes, pipette users, and liquid handling processes. This course is intended for laboratory managers and QA and QC supervisors. The course includes best practices in single and multi-channel pipetting technique, pipetting viscous solutions, pipette ergonomics, regulations and compliance, which includes reviewing the new and revised ISO standards, pipette repair and maintenance, comparing calibration methodologies. Participants will also learn how to diagnose pipette failure, and finally, using all of this to set up a quality assurance program. In this final case study, I'm going to take you through a situation where a lab performing endotoxin testing needed to prevent malfunctioning tests which were caused by faulty pipetting technique to prevent errors and contamination of samples. The training they received by Artel specialists ensured their technologists were using the correct and safe techniques to reduce errors and contamination. You can find additional case studies on our website, and you can certainly ask us if we have worked with any labs in similar situations to the one you're in. Thank you very much for your time listening, and now I'm going to pass the mic back over to Ryan for Q&A.
Well, thank you very much for that insightful presentation. I would like to invite our audience to continue to send their questions or comments right now using the questions window for this Q&A portion of the webinar. Now, as we move into the Q&A session, I'd also like to welcome Ryan Murphy, Technical Support and CS Supervisor for Advanced Instruments Artel Portfolio. He is one of the trainers that deliver the Pipette Technique certification courses and is joining us for the Q&A session. Also joining us now is Dr. Bjorn Karle, the Scientific Affairs Manager at Advanced Instruments. He has over 15 years of experience working with standards development organizations such as ISO, ASTM, and CLSI, and participated in the revision of the ISO 8655 series and the new development of the ISO 23783 series of standards. Thank you both for joining us today. I have already received some questions from our audience, so we'll get ourselves started with those. The very first question that I have here for you all would just simply like to know, is there guidance available on how to train pipette users?
That's a good question. We do have guidance available, Bjorn, I saw you were about to speak to. Like the slides mentioned, we do have training information available on our website. And then if you're more interested in having someone come on site, the services that Melinda went over as well.
Yeah, thank you, Ryan. I wanted to jump in as well. It's absolutely correct. There's plenty of guidance available. Melinda mentioned the 2 training seminars that we offer and the additional guidance that Ryan just mentioned on our website. As this guidance makes clear, there are really 2 components to trainings. It's the practical, the hands-on part of the training, but then also the theoretical approach to understand how pipettes work, what is necessary on how to pipette different liquids under different situations. And the first official guidance in an international standard was actually published earlier this year in the ASTM E1154 standard that clearly explains the training requirements for pipette operators. So that's an additional resource that you might find useful to consult.
All right, very good. That's very helpful. Thank you. The next question is wondering how often I should test pipettes between calibrations.
Oh, another good question. Let me take that one. In the overview of the standards, Candy mentioned our recommended calibration and test frequencies, but that's a good starting point. It's a good recommendation. And of course, some guidelines, some standards give minimum requirements, minimum time spans and frequencies for calibrations and routine tests. But it's important to understand that the calibration frequency and test frequency really depends on your own laboratory's risk assessment. And that really should be the foundation of developing the frequency for calibrations and routine tests. Remember, if you find a pipette, or that really applies to any device, if any device is found to be out of calibration at the time it is recalibrated, you will need to go back to the last time that device or that pipette was calibrated and was found in working order. So if you calibrate only once a year your pipettes, you will have to re-examine 1 year's worth of data. For every experiment, every assay, every result that this particular pipette was involved in needs to be re-examined, re-evaluated, and potentially invalidated. So it's important to have a pipette calibration and test frequency that is high enough to mitigate that risk. And in the standards world, we've been really going in the direction of risk management and risk assessment. You will see that most standards are moving away from prescriptive frequencies or prescriptive tolerances, but rather require an individual risk assessment by the laboratory for this individual purpose and individual situation. I hope that helps. That answers the question.
I think it does. That was very, very informative. Thank you. I have some more questions about calibrations. Someone's asking if I'm using different types of tips on one pipette, such as low retention or filter tips versus non-filter tips. Should I calibrate my pipettes with those tip types?
In short, yes. It's important to calibrate the pipette as it's being used, just to get a better reflection of the performance of that pipette. So if you're using low retention tips or filter tips in the lab, those are the tips you would want to use when calibrating.
All right, very good, thank you. Then another question for calibrations is, if I'm using a pipette in both forward and reverse mode, do I need to perform my calibrations using those modes?
I can I can answer that as well. Typically, pipettes are calibrated using the forward mode. That is the technique and the mode that is being used and prescribed in the calibration standards and calibration procedures. Now, that does not mean that you should not calibrate the pipette in reverse mode if you are often using the reverse mode. It is well known that the reverse mode includes or introduces larger errors in the volume delivery than the forward mode does, but it is important to understand those errors. So by all means, if you're using the pipette frequently in reverse mode, it would be worthwhile to calibrate or at least test the pipette so you know the error that's involved in that delivery mode.
All right, thank you. The next question here I have is wondering what types of tolerances should I designate for operator competency assessments?
I would say for the operator assessment tolerances, similar to the recommendation for the pipette calibration, ultimately your process will determine those requirements or those tolerances. So that's kind of the starting point, the broad recommendation. Bjorn, you may have more on that.
Yeah, that's absolutely correct, Ryan. The tolerances of the operator and the pipette work together. It is difficult to separate them. So if you think about it, you can use the most perfectly calibrated pipette, but if the technique of the operator is not up to par, that perfect calibration and functionality of the pipette doesn't really matter. It's the same thing with a musical instrument. You can have a perfectly tuned piano, but if you don't know how to play piano, it doesn't matter. There are some studies that have been conducted. Mostly, one study has been conducted by the Euramet organization. Euramet is the Association of the European National Metrology Institutes, and they looked at users' micropipettes and tried to see what is an achievable error tolerance. And if I'm not mistaken, the results can be interpreted in a way that about a 2% systematic error and a 1% random error is a good starting point, and it's typically achievable by users. And I believe that is also the recommended tolerances, or these are the recommended tolerances in ASTM E1154. So that might be a good starting point, but definitely your process tolerances, as Ryan mentioned, are the determining factor.
All right, very good. Thank you both for your thoughts there. The next question is wondering, can Artelware be used with a balance?
I can take that question. So the answer is yes. That's a short answer. A balance with an SICS6 protocol can be connected with Artelware and be used for pipettes larger than 5 milliliters. Now, if you are a user of Artelware already, or you're thinking about starting to use the software, we can provide a list of all the compatible balances.
All right, wonderful. Thank you so much for that, all of you, for all of your answers to the questions. However, we have reached the end of our time here today for the Q&A session. Now, if we couldn't attend to any questions, know that the team at Advanced Instruments will follow up with you. Or if you have some further questions, you can direct them to the email address that's up on the screen. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it helps us to improve our webinars. Now, I've also sent you a link in the chat box, and with this link, you'll be able to view the recording of this event on this page, and you can share this link with your colleagues when they register for the recording here as well. So I encourage you to do that. Now, please join me once more in thanking our speakers for their wonderful time here today. We hope you all found the webinar informative, Have a great day, everybody, and thank you for coming.
Thank you very much.
Thank you.